Hello, and welcome to RareOnAir, the Eurordis Rare Diseases Europe podcast series. I am your host, Julian Poulan, and in this podcast series, I speak to people from across the rare disease community who share their stories, their challenges, and their successes. In today's episode, I speak to Nick Sireau, a leading patient advocate and the Chief Executive Officer of the AKU Society. Nick's journey began more than 2 decades ago when his son was diagnosed with a rare genetic condition called alkaptonuria, or AKU. What followed was a deeply personal mission, one that ultimately contributed to the development and approval of a treatment that, quite remarkably, had its origins in a weed killer. I talk with Nick about the turning point that launched him into rare disease advocacy, his passion for drug repurposing as a way to deliver more treatments to patients, and his vision for the future of rare disease research and innovation. Nick, thank you so much for joining me today.
Very pleased to be here. Thank you.
Thank you. So my first question to you is, could you take us back to when your son, Julian, was first diagnosed with alkaptonuria? What were your initial thoughts upon that diagnosis? And how did that moment lead you to becoming a leading advocate for rare diseases? Can you tell me the story of that journey from diagnosis to advocate?
Of course, yeah. So it all started in 2000. So June was born in October 2000. And we brought him back from where he was born in the hospital. It was a Sunday. And we were changing his nappy and we noticed that they were red-black. So obviously we were very alarmed. And because it was a Sunday, we couldn't go and see our GP. So we called an emergency doctor who turned up and he tested for blood and didn't find any. And he asked what we'd eaten that day. And it so happened we'd had some red cabbage. So he said it was the dye from the red cabbage entering the breast milk and entering the baby and into the urine. So we were really dubious about that.
Can that happen?
No. Well,
Okay.
That's not as far as I know.
Yeah.
But it didn't sound very realistic. So next day we went to see our GP in Killick Street in London, and he sent off for a bunch of tests, and it came back 2 weeks later from Great Ormond Street with a diagnosis of this ultra-rare disease, alkaptonuria. So the first thing he told us is, whatever you do, don't go on the internet. And obviously that's exactly what we did. And we were very alarmed because we saw all these stories. At the time, this was the early days of the internet. And I don't think Google was around. I think it was Yahoo. And we came across all these stories of black bones, of joint deterioration. We joined a Yahoo group called alkaptonuria ochronosis. Ochronosis is the black pigment that happens in AKU. And all these patients were exchanging stories of severe joint deterioration and pain and how they were dealing with it. So we were really alarmed, alarmed.
What were the immediate repercussions of the diagnosis? Did you find that the healthcare was there, or did you rely quite heavily on these support groups and learning from the experiences of others in the newfound alkaptonuria community?
Yeah, so there wasn't really a support group. All there was a Yahoo email group.
Right.
And so there was really nothing, to be honest, apart from a few patients who were swapping stories. And when we went to Great Ormond Street, the doctors told us, look, there's nothing you can do.
Right.
They said that the NIH in the States was studying a potential treatment, but it was super early stage and they had no idea whether it could work or not. So they said, look, alkaptonuria won't affect your son till he's much older, so don't worry about it. Just give him a good childhood and stuff like that. Now, we were quite surprised, really, because we thought, well, that's not great. And actually, from what we learnt about it, it didn't seem at all as benign as they were saying. And so I got in touch with a patient in Liverpool, a guy called Robert Gregory, who had alkaptonuria, was in his 50s or 60s. And he wanted to set up a formal patient group. Okay. And so I went to see him in Liverpool and he invited me to join as one of the founding trustees. And this was in 2002. And so I did. And also he was in touch with a doctor in Liverpool called Dr. Ranganath.
Right.
And Robert Gregory, who we called Bob, had convinced Ranga, as we called him, to join as a founder of the AKU Society. And Ranga had said yes.
Oh, cool.
And that turned out to be probably the best decision ever made for the AKU community because Ranga is really the hero, scientific and clinically, of the AKU community, who has really driven everything forwards.
I see. And so what was your initial involvement in the AKU Society upon it founding? Could you tell us the story of those early days of the AKU Society coming into existence?
Yes, so we set up in 2002. We registered it as a charity in 2003. And then my
Right.
second son, Daniel, was born. And I spent the first few days quite obsessively checking his nappies to see if they went red-black. And after a few days, they did. So I knew he had alkaptonuria, and then that was confirmed and stuff. So I started putting a lot of energy into alkaptonuria, and my scientific knowledge was very rudimentary. And basically what would happen is that Bob and I would go and see Ranga in Liverpool and ask him, what actually do we need to do? And fortunately, Ranga really had a really good plan.
Right.
And so the first thing he wanted to do was actually do a post-mortem, an autopsy of an AKU patient who had recently passed away and who had donated her body to medical science for the better understanding of alkaptonuria.
I see.
So Ranga said, look, we need £5,000 so that we can actually pay for the autopsy. So the first thing I did was run a half marathon and raise funds from family and friends. And that then paid for the autopsy and all that, which was done by a doctor called Tim Helliwell, a pathologist, as well as Ranga. And then that was published. And that was the first time that anybody had ever had actually seen what was happening inside the body of a patient. And it was really surprising because what we saw was certain parts of the body were very pigmented, very black, and others weren't.
Wow.
And we were kind of wondering, why is that the case? And you have to remember that alkaptonuria was the first disease to ever be shown to be inherited.
I think I remember this. Is it something to do with a mummy?
Well, there is an Egyptian mummy at the University of Chicago that has evidence of alkaptonuria, but it was in 1901, a scientist, clinician in London called Sir Archibald Garrod, who first identified that alkaptonuria was inherited. And you have to understand that until then, people didn't know that diseases could be inherited. They knew that you could have contagious diseases, but he was the first one to actually prove that a disease could be inherited. So it was totally groundbreaking.
Oh, wow.
And
I would have thought that even evolutionary science would have got underway by then.
No.
I'm surprised.
Well,
that
there was the theory of genetics. And so there was Gregory Mendel, the Swiss monk, who a few decades before had been studying peas and all that, and had developed this theory of Mendelian inheritance. And Sir Archibald Garrod applied that to alkaptonuria. So he was decades ahead of his time. And he did these Croonian lectures, I think in 1906 or 1908, about these inborn errors of metabolism. And alkaptonuria was the iconic one. So it was actually the first disease to be shown to be genetic or inherited. So it's on all the medical history books. But because it's so rare, it took so long for actual research really to pick up.
And so what did this autopsy reveal, or what did it allow to be revealed? And in what ways did the autopsy itself become something that would advance understanding of alkaptonuria and the science around it?
Yeah, that's a good question. So what then happened is Professor Ranganath started working at the Royal Liverpool University Hospital, and he basically introduced us to a professor called Jim Gallagher. And what Jim really wanted to look at was this whole pigmentation at the cellular level. How does it happen?
Yeah.
So a lot of that thinking came out of this kind of, autopsy and stuff. And so what we then did is, again, I did some fundraising and we raised funds to pay for a PhD program, basically. So a young PhD called Adam, who then spent 3 years really trying to understand, how the culprit molecule. So AKU is a monogenic disease. It's due to a deficiency in a single gene. And we now know there's hundreds of mutations in this gene that lead to alkaptonuria. And the gene basically codes for an enzyme called homogentisate dioxygenase. And the HGD enzyme is on the pathway of the breakdown of tyrosine, which is an amino acid. And because people like my boys have this genetic mutation, they can't break down. Basically, the enzyme malfunctions or isn't even created in some cases. And so they can't break down an acid called homogentisic acid, that we call HGA. And that HGA accumulates at 2,000 times the normal rate.
Wow.
And it gets excreted in the urine and then turns black in contact with air. Or in parts of the body in this process called ochronosis. So what Adam did in his PhD was really showing that in cell models, when you added the homogentisic acid, it led to this black pigment and all that. So the HGA really is the culprit molecule. And that really helped us understand that to treat alkaptonuria, you really just had to lead to a significant reduction in this acid to near normal levels. And then you'd be able to basically slow down or prevent the evolution of the disease.
I see. And actually, can you recap for us, what exactly are the symptoms of the disease? How does it progress? In what ways does it impact one's life? How does it impact one's life?
So the main symptoms, apart from like the urine and all that, are very severe joint deterioration.
Right.
So it's called alkaptonuria AKU, but it's also known as black bone disease because the bones literally go black and brittle. And what happens is that in early adulthood, patients will start to have a joint problems with their back and all that kind of stuff. And that will get steadily worse. We did a number of studies and we found that around a third of our patients get diagnosed at birth, like my boys did. But 2/3 don't. And that might be for reasons such as, nappies are very absorbent. We were using washable nappies. That's why we saw the black pigment. But some people just don't see it. Or patients, the parents will just believe what the doctor tells them, which is like, oh, it's, like we've had some patients, the parents were told, oh, don't worry, it's nothing.
Yeah.
But so generally what then happens is...
But surely it persists as a problem.
It persists, yeah. So the black urine persists, but they just think it's normal or they ignore it. And then what happens is the patients then go on this diagnostic odyssey, and it might take 10 to 15 years.
Yeah.
So we've had patients who have spent years going from one doctor to the next, some even being referred for psychiatric treatment because the doctors don't think there's anything wrong with them before they eventually get a correct diagnosis. So it's joint deterioration. All the joints in the body will start to get affected. And we have one patient who has had 11 joint replacements. So every major joint has been replaced.
And how immediate are the symptoms that actually impact well-being and health?
It takes years for them to really develop.
Right. How detrimental is a late diagnosis?
It can be pretty catastrophic, a late diagnosis, because it's not really reversible. Now, what we have shown is that the drug nitisinone can partially reverse, at least we think so. But to be perfectly honest, the best it can really do in advanced stages is slow the progression of the disease. Right. And that's because the joints are already so damaged that, so a late diagnosis is better than nothing, but it's not great.
Okay. Well, you've mentioned for the first time in our discussion nitisinone. Can you share the story of this molecule? How was it discovered and how was it ultimately developed into a treatment for your sons and others living with AKU?
So it's quite an extraordinary drug, nitisinone. It was originally invented in the 80s by a scientist who was gardening in his garden, and he had an Australian bottlebrush plant, and he noticed there were no weeds around it, and he was very curious, and he eventually identified the compound in the plant that was killing the weeds, and he synthesized it. And they started to develop it as a weed killer, nitisinone. And it's part of a group of molecules called triketones. Okay. And in the end, nitisinone was never commercialized as a weed killer because it tended to be too potent. Okay. Because it was so effective. But an analog of nitisinone called mesotrione is one of the world's most successfully commercial weed killers. But what they found was that nitisinone worked in plants, the same thing as in animals and in humans, and it's an enzyme inhibitor. Nitisinone. So it inhibits one step up from the homogentisate dioxygenase that causes all the problems in alkaptonuria. And the reason it works as a weed killer is that it prevents photosynthesis. Basically. And so through lateral thinking, there was a scientist there called Ted Locke, who's one of our advisors now, and worked on the tyrosine pathway in rats. And he realized that there was a human condition called hereditary tyrosinemia type 1. HT1, that's an ultra-rare disease. And so he thought, if nitisinone works like this in rats, it should work in a similar way in humans. I believe the story is that he contacted the Karolinska Institute in Sweden. And in the early 90s, the Karolinska Institute basically gave the weed killer to babies with this rare disease, HT1.
Oh, wow.
Now, HT1, untreated, kills children with liver cancer by age 2. And when they fed the weed killer to the children, 30 years later, the kids are still alive.
Wow.
So really quite extraordinary. And then what happened in the late 90s, there was a group at the National Institutes of Health in the US led by Dr. Bill Gall, who said, maybe we can use this drug nitisinone for alkaptonuria because it's on the same metabolic pathway.
Right.
And they tried really hard. They did a clinical study 20 years ago, and that failed. And the reason that failed was because the American regulators, the FDA, insisted on a single clinical endpoint, which was hip rotation. Right. And they said, we want to see an improvement in hip rotation in patients on this drug. And unfortunately, AKU affects patients very differently. Some it's the elbows, some it's the hips, some it's the knees and all that. And the study was only on 20 patients on the drug and 20 patients off the drug, and it didn't last long enough, so they showed nothing. And that nearly killed the whole program. Jesus. So that's when we stepped in and decided to do a much larger study, to really prove that it worked.
Right. Do you know if, at least in America, the ways that clinical outcomes are measured have improved?
There's been some significant developments. I know that nitazoxanone is going through the FDA approval process at the moment, and I believe the FDA has become more flexible in its approach, but we'll have to see. So what happened was when the NIH trial failed in 2009, which was devastating for the community, as well as the scientists. The scientists are a really strong bunch of people at the NIH. But the community was devastated because a lot of patients on the study said that they really felt an improvement.
Wow.
And Professor Ranganath basically said he thought that it would be possible to actually do a study that would work.
Okay.
Because he had developed by then what was called a composite endpoint, which measured every aspect of AKU. So the eyes, the ears, the heart, all the different joints and all that, and came up with a single score that you could measure over time to see the evolution of the disease. So really good thinking. And to do that, the AKU Society had raised some funds to finance a natural history study. So anyway, we put together a consortium in 2010, so 13 partner organisations. Liverpool was leading it, but we had the Hôpital Necker in Paris, very strong on rare diseases. And also we had the Slovak National Institute of Rheumatic Diseases, because there are many patients in Slovakia, actually.
Oh, wow.
And we managed to convince the company that owned the drug, Sobi, to join the consortium. And I think what really clinched it was we then applied for European funding, so an FP7 grant. And out of 500 applications, we came first.
Wow.
And we got 6 million euros from the EC. Oh, wow. No other funding body provides any funding on that level. So we're hugely grateful to the European Commission for this. And that allowed us then to put together this 7-year program from 2012 to 2019, where we did a phase 2 study, basically, on 40 patients to look at which dose worked the best at reducing the homogentisic acid. And we found that the dose of 8 milligrams reduced it by, I think, 99.7%.
Oh, wow.
And then we went to a 10 milligram dose, because that was an actual single pill, to the phase 3 study, which was apparently the largest ever study of an inborn error of metabolism, on 139 patients, over 4 years, with these composite endpoints and all that, with patients from across Europe and the Middle East. And that went exceptionally well. And I think one of the reasons was, well, Professor Ranganath just led it exceptionally well.
Yeah.
Secondly, the consortium worked really well, and the partnership with the pharma company Sobi worked really well. Thirdly, the patients were super motivated. I think also the support of the patient group, AKU Society in the UK and ALCAP in France, really helped. And also, we had very good scientific advice from the European Medicines Agency in November 2012.
Oh, cool.
When we went to see them, they said, look, what we want you to do is to put the homogentisic acid, the culprit molecule, as the primary endpoint, And as secondary endpoints, the composite endpoints and all that, we just want to see a positive trend.
Okay.
Not even necessarily statistical significance.
Oh, okay.
And they said the reason for that is we understand that alkaptonuria is super rare. It's 1 in 500,000.
Okay.
So it's very difficult to do. That it's a slowly progressive disease.
Yeah.
And that, we have to really take that into consideration when evaluating the study.
Interesting.
So they were much more flexible and I would say progressive and forward-thinking than the FDA had been, just a few years before.
Yeah. And
that really helped. In the end, our primary endpoint, we just blew it out of the water because the homogentisic acid was reduced by 99.8%. Wow. So an amazingly effective drug. And on top of that, the secondary endpoint, the composite endpoint, did reach statistical significance.
Okay.
So it was better than we could have dreamt of. And when Professor Ranganath and Sobi went over all the data in 2019, they were really pleased. And so Sobi then applied to the European Medicines Agency, put through the process, and the EMA gave a positive opinion in September 2020, and a month later, the European Commission gave marketing authorization.
Wow. It's an impressive
It was
story.
really good. Really good. And I think a lot of things came in place really to make it work.
Yeah.
And it's an effective drug. And in parallel to that, in 2012, we had also got funding from NHS England's Highly Specialised Services Department, which is the department that funds ultra-rare diseases, to set up a national treatment centre at the Royal Liverpool University Hospital. This was the first ever in the world national treatment centre. And that has now been running for 13 years.
Wow.
And what that did is that it meant that all patients with alkaptonuria in the UK could go to Liverpool once a year for 4 days of tests, MRIs, X-rays, everything you can imagine, and they could already access the drug off-label.
Right, okay.
So in parallel to the clinical studies we've done thanks to EC funding, there's been this off-label study happening that has proven exactly the same thing, that nitisinone, if taken early enough, prevents the disease. Otherwise, it halts the progression, or if taken later in life, slows down the progression of the disease.
Wow, that's brilliant.
And has a good side effects profile, a good safety profile.
Yeah. And do you know if either nations in Europe or even around the world have an approach that is similar to that of the UK, of having one specialist base that the national disease population can benefit from in terms of receiving specialised healthcare?
I'm not aware of exactly the same model. I think in other countries, I think in France, you will get these reference centres that I think pull together rare diseases. So it's a different format. But I think French patients with alkaptonuria get good support and all that. The sad story is the US still hasn't approved it, although it's currently going through the FDA approval process. But American patients really do struggle. And then it comes up to their private insurance, whether they will give them off-label access and stuff. But around Europe, I think just about every single country now provides access to nitisinone for alkaptonuria, apart from, I think, Romania, last time I checked, where we're still trying to get there. But then after the EMA approval, there was the whole reimbursement process to go through, which was patchy.
Yeah.
Because that's by country by country.
Is this the same thing as like health technology assessments and so on? Yeah.
And we got stuck in France with the Haute Autorité de Santé, so that had to be appealed, and now it's okay. In Italy, similar thing. In the UK, it became very difficult because you have England, you have Wales, you have Scotland and all that, but that we managed to get through all of that and sort that all out. And then other countries, India approved it a couple of years ago, and I think there's a generic company in India providing it, so that's good news. And there's a few countries around the world that have approved it. But one of the big things we're working on now is we set up a year ago an international patient registry for alkaptonuria.
Okay.
And that is really to try and help patients around the world gather data about their illness and help them get access to nitisinone locally.
Brilliant.
Because at the moment you have this situation where Europe, the UK, and a few other countries have access to it, but others are still trying. And I think the most frustrating is it's a really cheap drug now. Because it's gone generic, I think a 2 milligram dose, which is what we've been originally were treating people with, is just a couple of euros a day. So
of course,
from a monetary point of view, shouldn't be much of a barrier in access to it.
What I find interesting is what you've said about it not yet being approved, right, in America. Do you think that the difference in the fate of nitisinone up to today between Europe, the UK, and on the other side, America, is it to do with a difference in regulation or a difference in, or a fortune within Europe about the advocacy that you and your partners could successfully execute it? If that makes sense.
Yeah. I'm not sure, to be honest. I think there's some systemic issues probably in the US that have made it more difficult. I've been told that the FDA has evolved in its thinking, particularly to do with rare genetic diseases.
Yeah.
So I'm hoping that will be positive. But I think at least 20 years ago, they did not have the kind of more progressive thinking that Europe had. So hopefully things have moved on and all that. I'm not sure, really. One of the issues you get, we were fortunate because Sobi really came on board and for 7 years were a full partner in the program. And they put quite significant of their own resources and time into this, and all that. But the problem you get when a drug becomes generic is that it's very difficult to have a commercial incentive to actually develop it, particularly when the drug has become so cheap.
Of course.
And that is a much wider problem, I think, to do with intellectual property laws and all that kind of stuff. And so, we were fortunate in that we could bring everything together in the EC funding to really make it all work, at least in Europe.
Yeah. So actually, of course, you've given the case study of nitisinone as a drug that was repurposed. Could you reiterate what exactly drug repurposing is? You've mentioned the difficulties with grant funding already, but could you also share with me, upon re-explaining what drug repurposing is, what you think are the main challenges or barriers to successful drug repurposing?
Yeah, so drug repurposing is a very simple concept. You basically look at an existing drug that has been approved for one disease, and you look at how to reuse it for another disease. And the concept is quite simple because the average drug will work on dozens and dozens of different mechanisms in the body. So you take like beta blockers. They are used for anxiety, but apparently, they are used also for post-traumatic stress disorder.
I see.
And that's because they affect, all kinds of different mechanisms within the body. The beauty of drug repurposing is that if a drug has already been approved, it means, it's gone through all the safety testing, the clinical studies and all that. So in theory, it should be quite easy to then redevelop it for another condition.
Yeah.
The problem you then face is multifold. The first one is, where do you get the money?
Yeah.
To actually prove that it works for a new disease. And to do that, you have to do clinical studies. And the clinical studies you have to do are often quite large.
Yeah.
You're thinking tens of millions. Where do you get the money from to do that? So you can apply for grant funding bodies, but in the UK, the MRC or the NIHR will not give you 10, 15 million to repurpose a drug.
Yeah.
So then you have to go looking for commercial financing. And none of the commercial investors will give money if they don't think there's a return. So it just doesn't work commercially either. So you have all these drugs that could be used for other diseases, but because of systemic reasons, they're just not being developed.
What do you place more emotional investment in as a way of addressing these challenges? Do you think that the way to try to make sure that drug repurposing is more widespread and embraced, is it more finding ways to work within the current infrastructure, or is it more pushing for change with the structure itself?
So you have to do both. Now, as an individual patient group working on one disease, your focus is to try and treat that disease. So you have to work within existing structures.
Yeah.
But what I try and encourage all patient groups to do is give a little bit of their time to the whole community, because the work we've done over the past 25 years in alkaptonuria would not have achieved much without the groundwork that groups such as Eurordis, European and globally, and also, say, Genetic Alliance in the UK. What these groups do is that they do the groundwork and set up the system and the policies that then help individual patient groups really make things work. Yeah. So I think there is a need for systemic change. And 13 years ago, I set up a charity called Find a Cure, which is now called Beacon for Rare Diseases. And Beacon works on two things. One is helping kitchen table groups in the UK, and the other thing is drug repurposing. And Beacon is part of Remedy for All, which I think Eurordis is part of too, which I just think is an absolutely fantastic initiative. And Remedy for All is looking at what do we need to change in the system to make drug repurposing work better. And I think some of the things are regulatory changes. So if you expect a bunch of some academics or a small biotech to manage to repurpose a drug to exactly the same regulatory standards as a common disease or as a completely new chemical entity or whatever, it's going to be a struggle. Yeah. Because they won't manage to get the money. So you need to somehow rethink the regulation so that you still have good evidence. Yeah. I mean, the safety is non-negotiable.
Of course.
Absolutely. But there needs to be some regulatory framework that means that the barrier to entry is kind of lower, for these repurposed drugs, if you see what I mean. So that the level of financing you need becomes much more reasonable to actually get it done. So I don't know, I'm not especially in regulatory affairs and all that, but I think also the whole intellectual property needs to be rethought. Right. Because at the moment, once the patent runs out on a drug, I mean, there are ways that companies can extend their patents, but once all that runs out, that's it. Yeah. The drug's generic. And the fact the drug goes generic is fantastic from a financial point of view for the health systems because they become much cheaper. Yeah. But from an incentive point of view to repurpose them becomes difficult. So I don't know. I'm not an expert in these areas, but there must be ways of changing these things to increase the incentives for people to develop them.
So speaking beyond drug repurposing, are there any areas of research that you're keeping a close eye on? For alkaptonuria, do you think the science has further to progress? And for rare diseases more generally, what are you kind of keeping a close eye on in terms of and investing some hope in with regard to the delivery of more treatments?
So my real area of specialism is alkaptonuria. And we've looked at a number of other therapies over the years. We've looked at gene therapy. The problem with gene therapy, particularly with the kind of AAV viral vectors, is the immune reaction, that many people are just naturally immune to them. And the fact that there's been a number of deaths also in gene therapy trials over the years. So for a disease such as alkaptonuria, where there's a very effective existing treatment, a gene therapy would have to be 100% safe. And I mean, not 99.9% safe, 100% safe.
Is that possible?
Exactly. For us to even consider So we've looked at other small molecules. We've looked also at therapies to go with nitisinone, because nitisinone, whilst it's a good therapy, is far from perfect.
Okay.
So what happens, because it's an enzyme inhibitor, you get a rebound effect where tyrosine goes through the roof. Okay. So my boys have to go on a low protein diet to keep their tyrosine levels down, because otherwise, in some cases, they can have corneal crystals and kind of problems with the eyes and rashes and stuff. So they're constantly monitored for their tyrosine levels. And we've been working with the University of Liverpool and with a couple of pharma companies on tyrosine inhibitors.
Right, okay.
And we've got some really good data on that, and that would be co-therapies with nitisinone, But also we've looked at mRNA therapy. And mRNA therapies could potentially work because the mRNA can basically code for the missing enzyme.
Oh, wow.
So we've done some preclinical work on that and all that. So I think mRNA therapy is quite interesting for rare diseases.
Yeah.
There are some problems. So at the moment, the mRNAs, the mRNA might only last for, the effect might only last for a day or two.
Yeah.
So how do you actually increase the effect so that people are just getting a dose like once a month or stuff like that? So there's still a long way to go in mRNA therapy, but I think there's some quite promising work happening there. And the great thing with those kinds of therapies is that they can then be used for many rare genetic diseases.
Yeah, I see. So my final question to you, of course, you've been such a huge part of the immense success of nitisinone being approved. And you've made a great difference for patients, of course, with many partners. But what's next for you personally, whether with the AKU Society or through Beacon Rare Diseases? Do you have any other new kind of rare disease projects on the horizon?
Not in rare diseases, but in mental health.
Oh, really?
Yeah. So, again, it's a very strong personal reason. I work on obsessive compulsive disorder, OCD.
Oh, right.
So I've had OCD for about 35 years, sometimes very seriously. And I run a support group in Cambridge in the UK of people with severe OCD. And one of my best friends from my best friend from my support group 5 years ago took his own life. And he had a form of OCD called false memory OCD, where his brain would make up false memories of horrific crimes.
Oh, wow.
And then he would obsess about had he done it or not. And it drove him to take his own life. And OCD is very common. So unlike rare diseases, OCD is at least 3% of the population.
Yeah.
And the problem with OCD is that, again, there's a misconception. Most people think it's about lining up your pencils and colour-coding your spreadsheets. And it's got really nothing to do with that. It's a devastating mental disorder. So in the UK, 1.5 million people suffer from it. So I set up a foundation 7 years ago called Orchard OCD that funds academic research.
Okay.
And we have funded a study of a psychedelic, psilocybin, at Imperial College under Professor David Nutt. That went well. That'll be published soon. We've funded a study of neuromodulation OCD. We have funded a study of drug repurposing, a drug called tolcapone at the University of Chicago, which is looking promising.
That's really interesting. And I wish you the very best of luck with your endeavours in the area of mental health. Yeah. Nick, it's been fascinating learning about the story of nitisinone, the history of science in alkaptonuria, all the ways in which drug repurposing can be further embraced. Thank you very much for talking with me today.
Thanks for having me.